Assalamu alaikum. Today is the third lesson in the series of A to level biology topics, and we have been doing chapter number 14, which is homeostasis. We have already covered negative feedback, thermoregulation, as well as control of blood glucose concentration. Today in this lesson, we will inshallah cover osmoregulation. So for that, we will inshallah describe deamination of amino acids and outline the formation of urea and the urea cycle. Now your syllabus says biochemical details of the urea cycle are not required. So you don't need to worry about that, right? Then describe the gross structure of kidney and the detailed structure of nephron with its associated blood vessels using photomicrographs and electromicrographs. You're also required to describe how the processes of ultrafiltration and selective reabsorption are involved in the formation of urine in the nephron. And lastly, we'll cover the role of hypothalamus, posterior pituitary gland, antidiuretic hormone, collecting ducts in osmoregulation. Okay, so let's start with the control of blood water potential, or in other terms, osmoregulation. So to understand the process, we need to first discuss the process of excretion. So you know that there are many metabolic reactions taking place in the body, and that means a lot of waste products are also being formed. So the body actually needs to remove these waste products. And as it removes these waste products, the process is known as excretion. So there are many excretory products formed in the human body, but there are two of particular importance. One of them is carbon dioxide, and the other one is urea. And both of them are actually being made in very large quantities as compared to others. Now, carbon dioxide is produced continuously by the cells that are respiring aerobically. The waste carbon dioxide is transported from the respiring cells to the lungs in the bloodstream. Then gas exchange occurs within the lungs, and carbon dioxide diffuses from the blood into the alveoli. And then it is excreted in the air as we breathe it out. However, urea is actually produced in the liver, and it is produced from excess amino acids. Like you've eaten a lot of protein, but definitely like all of it cannot be stored in the body. So what happens? Body can actually still use amino acids within those proteins and use it as a source of energy, right? To make this energy accessible, what happens is that an amino group is removed from each amino acid along with one extra hydrogen. So these two combine to form ammonia, right? The process is known as deamination, right? So I just start again. So what happens? Amino group of an amino acid is removed together with an extra hydrogen atom, and these two combine to form ammonia. The remaining keto acid that is there, it actually enters the Krebs cycle to be respired, because we definitely need to make energy out of it, and then it can be converted into glucose, or it may be converted into glycogen, or it may be converted to fat for storage. So these are the two steps in the process of formation of urea. So here you can see this is an amino acid, and you can see amino group, one extra hydrogen. These two are removed to make ammonia, right? And this is the keto acid that remains. It can enter the Krebs cycle or convert into glucose or fat. And later on, ammonia actually combines with carbon dioxide to give urea as well as water. Now, the thing with ammonia is that it is very soluble and it is highly toxic, right? So we actually have to quickly remove it from the body. So what happens is that it is actually converted into urea, which is less soluble as well as less toxic than ammonia. It's converted immediately into urea. And then, as we just discussed, yes, how it happens, ammonia actually combines with carbon dioxide to form urea, and then urea is excreted out from the kidney. So urea is the main So urea is the main nitrogenous excretory product of humans, right? The main. We also produce a small quantities of other nitrogenous excretory products, and two of those are creatinine and uric acid. Now, creatinine actually is made from a substance which is creatine, which is made in the liver from certain amino acids. So what happens? Much of this creatine is actually used as a stored form of energy, right? So it's there in the muscle in the form of creatine phosphate, and it acts as an energy store. However, some of it is actually converted into creatinine as well. So this creatinine is excreted to the kidneys, right? But uric acid is actually made from breakdown of purines. Okay? Purines are adenine and guanine from the nucleotides, and uric acid is not made from amino acids. This is something that you need to remember. While urea, as it is formed, it diffuses from the liver cells, then it comes into the blood plasma, and then it actually gets excreted through the kidney, right? And like all of the urea that actually is made, it has to be excreted, right? Because again, it's like toxic. So the body doesn't need to actually get it built up. It actually has to get it removed from the body. So to understand this whole process of excretion, you need to first understand the gross structure of kidneys. So if you look at this picture here, it is actually taken from the back. So just to show you that kidneys actually they lie retroperitoneally. Retro means behind. Peritoneum is actually a lining that covers these abdominal viscera. So it's a protective layer. So you can see two kidneys on either side of the vertebral column. So this is the vertebral column that you can see. So these are the thoracic vertebrae named as T10, T11, T12, and the ones started from L are the lumbar vertebrae, right? So a typical kidney actually is around 3 vertebrae long, like its length is like that. And so it ranges from T12, the 12th thoracic vertebrae, to the third lumbar vertebrae, right? And the right kidney is a little lower as compared to the left one because of the liver, right? So kidneys, if you look at the shape, they are bean shaped. Their color is reddish brown. The medial border, this part if you see is a little concave, while the other one is a little, the outer border, the lateral one is a little convex, right? And you can see a hilus there in the middle. So for this hilus, actually, the renal arteries, the renal veins, and the renal nerves, they enter. So actually, this is known as a hilus or hilum. Then each kidney is actually covered by a capsule, okay, a protective layer, which forms a fibrous tissue. So the outer layer is capsule. And then if we look at the microscopic structure, it is divided into an external renal cortex and an internal renal medulla, which we are going to look at in the next slide. And then if you look at the medulla, it is made out of cone-shaped renal pyramids. And then each kidney may contain around 8 to 18 renal pyramids. We'll just have a look at it in the next slide. So say these are the few structures that you actually required right now as far as your A-level syllabus is concerned. So this is the capsule, the one outside that you see, the fibrous protective sheet. Then you can see this is the renal cortex, this part here. This is the cortex. And then this is the medulla, the inner part, this thing, this entire part is the medulla, which contains a renal pyramid. So you can see this is one pyramid here, another pyramid, another pyramid, another pyramid, right? So these are the big pyramids which actually made up the medulla, right? Then you're also required to know about the renal veins, the renal arteries, right? And this here is actually renal pelvis. So all these medulla, right? So you can see the collecting ducts actually will join there. This is the renal pelvis, and then this is the ureter. All of this connect to form the ureter, which goes down and joins the urinary bladder, and then the urine goes out through the urethra. So when we talk about the microscopic structure of the kidney, so each kidney actually is made up of uriniferous tubules. These uriniferous tubules actually are divided into two parts. So one is known as nephron and the other is known as the collecting duct. Nephron is actually the main functional and structural unit of the kidney. And each kidney is said to have around 1 million nephrons, right? So what happens, along with that, a small number of connective tissues, blood vessels, nerves, lymph vessels, they're also present within the uriniferous tubules. So we'll just have a look at the picture in the next slide. So here you can see the structure of nephron. So if you focus here, the structure that you see is known as the Bowman's capsule. So this is an enlarged version. So this is structure that you see here, this part. This is known as the Bowman's capsule, right? It contains these capillaries, these twisted capillaries, which is known as a glomerulus. So what actually happens is that the renal artery actually divides into arterioles, and then the name that's given to is afferent arteriole, right? So afferent arteriole actually goes into the glomerulus. Then these capillaries, they actually start to narrow down also, and then once it goes out of the glomerulus, it is named as efferent arteriole, right? So you might be noticing a little difference in the width of the afferent arteriole as well as the efferent arteriole. So it actually, according to that, there's a difference in the functions also. We'll talk about that in a little while. Okay, but just to understand, the Bowman's capsule contains the glomerulus, and together these two, the Bowman's capsule along with the glomerulus, are also known as Malpighian body, right? They're also known as Malpighian body. Bowman's capsule plus the glomerulus. Then you can see that actually it starts to twist up into a proximal convoluted tubule. So the first part that you see here, the one part, it actually starts to twist up. This part is known as proximal convoluted tubule. And then you see actually it descends down into a loop of Henle. So this part here is known as the loop of Henle. And as this part goes down, so it is known as the descending limb of loop of Henle. and this part goes up. So this is known as an ascending limb of loop of Henle. So here you can see some structure differences here also. So definitely the functions actually differ in that way. And then you can see the ascending limb of loop of Henle then again goes up and again becomes convoluted. So this time it is known as a distal convoluted tubule, right? And then it again goes down, and which is then known as a collecting duct, which then later on opens into the pelvis. So you might also be noticing that some part of the nephron is actually found in the cortex, while some part of it actually is found in the medulla. So the glomerulus, Bowman's capsule, the proximal convoluted tubule, and the distal convoluted tubule actually all lie in the cortex, while this the loop of Henle, the descending limb of loop of Henle, the ascending limb. Now this part here is known as a proper limb of loop of Henle, along with the collecting duct. These are all found in the medulla, right? So this is how these are present. So here you can see in this diagram as well. Since this part here is a cortex, let me just mark that. Okay, so you can see the Bowman's capsule, the glomerulus, the proximal convoluted tubule, the distal convoluted tubule, right? So these all are there in the cortex. While if you look at the loop of Henle here and the collecting duct here, they are found in the medulla. So we've already discussed the structure of the nephron, but just quickly, let's just go through these. So each nephron is a tubular structure, as we just saw in the previous diagram. The free end is closed and the other end opens into the collecting duct. So we saw that also, right? So that's the free end, right? It's enclosed, and the other one actually opens into the collecting duct, as we have seen. And these are the structures. The Bowman's capsule, along with the glomerulus, together will be known as Malpighian body. Then comes the proximal convoluted tubule. Then comes the descending limb of loop of Henle. Then comes the proper limb of loop of Henle. The one that you saw just like that, this one, the proper limb of loop of Henle. Then comes the ascending limb of loop of Henle, and finally comes the distal convoluted tubule, right? So here you will see that the entire nephron is actually surrounded by a lot of capillaries known as peritubular capillaries. So these capillaries, a lot of exchange takes place between the nephrons and the blood plasma. So things that have to be excreted out actually gets absorbed into the tubule, into the lumen, and the things which have to be reabsorbed back into the blood actually connected to these capillaries. So depending on every structure, the function differs, and we'll talk about them one by one. So now let's talk about the basic function of nephron, which is filtering and reabsorption. So as we talk about filtering, so basically we're talking about ultrafiltration. And when we talk about reabsorption, we're talking about selective reabsorption, because everything does not get reabsorbed. Only selected substances get reabsorbed from the filtrate. So the basic function of the nephron is to filter the blood, to remove waste and selectively reabsorb. So make sure you're looking and using this word constantly, selective reabsorption. Please don't use the term absorb only, right? So use the substance back into the blood, and because it's actually making urine. The urine contains these, it contains water, it contains dissolved salts, it contains urea, right? It contains other small substances such as hormones and excess vitamins. However, what urine does not contain is actually protein and blood cells. The reason being, proteins and blood cells are way too big. So they cannot be filtered out of the blood, right? And glucose is actually, all of the glucose is reabsorbed in the proximal convoluted tubule. So you will not be able to see any glucose in the urine, right? So it might contain every other thing, water, dissolved salts, urea, but it will not contain any protein and blood cells because of the size. And it would not contain any glucose because all of the glucose, 100% of the glucose is reabsorbed in the proximal convoluted. So here you may see all the concentration of proteins, amino acids, glucose, urea, mineral ions in the blood plasma, as well as the concentration in the filtrate, right? And then finally, we'll actually compare with the concentration in the urine. This is how it happens. In the blood plasma, we have around 80 gram per decimeter cube of proteins, 0.5 amino acids. So you can see all the values. You see in the filtrate, every value is exactly the same except for proteins. And as we just discussed earlier, this happens because proteins are too big to be filtered into the glomerular filtrate. But all of the other molecules, right, they are passed as they are, right? But if you look at the concentration of these substances in the urine, so you see that proteins, a very, very small amount of protein can go out, right? But you can see there is no amino acid in the urine. There is no glucose at all. And even if you look at the mineral ions, their concentration actually has reduced. But if you look at urea, the urea's concentration has increased. The reason behind that is that, well, see, like there is no ammonia now, there's no glucose, so the relative concentration of urea actually seems seems actually more as compared to the others, because others are not there anymore, right? So the formation of urine actually takes place in three major steps. Now, the first two, glomerular ultrafiltration and tubular selective reabsorption, are of the most important. Now, while besides reabsorption and filtration, some secretion of substances also takes place, so that is known as tubular secretion. So it happens at different parts of the nephron. But again, the most important that we'll be dealing with is glomerular ultrafiltration as well as tubular selective reabsorption. So in the previous video, we talked about excretion, the role of liver in excretion. Then we talked about the gross structure of the kidneys and the structure of the nephrons. So now we'll be specifically talking about the function of nephrons in formation of urine. So this takes place in three major steps. The first and foremost is the glomerular ultrafiltration. Then comes the tubular selective reabsorption, and then comes the tubular secretion. So glomerular ultrafiltration is a process by which different substances are filtered, which are present in the blood, and they're filtered according to difference in their molecular weights under very high pressure. And these are filtered through pores in the capillaries of the glomerular wall, as well as through the pores in the inner wall of the Bowman's capsule, right? And this forms a glomerular filtrate. So this whole process is known as glomerular ultrafiltration. So here you actually can see the structure modifications that actually make it happen. So here you can see these are the capillaries in the glomerulus, and these capillaries have endothelial linings which have many tiny perforations in it. So this is actually an enlarged version. So you can see this is a capillary, right? And this is definitely a blood cell. And these endothelial lining, you can see they have many small perforations in them, right? Then there's a second layer which is known as a basement membrane. So the one that you see in the middle. So this is a basement membrane that acts as a filter. And the third layer is actually the inner lining of the Bowman's capsule, and which actually has many finger-like projections, and these are known as porocytes. Now, these porocytes also have gaps between them, right? So the water content and contents such as glucose, amino acids, ions, they can actually pass through these perforations in the capillaries, then they're filtered out, and then they go into the Bowman's lumen, right? But then protein particles as well as the blood particles, they are not able to pass through because of bigger size. So through these tiny holes, only glucose, amino acids, and water can pass out into the lumen. So that is what is known as glomerular filtrate. So we just discussed about the structural modifications here. So we talked about two cell layers. One was endothelial layer of the capillaries. The other was the inner wall of the porocytes, the cells of the Bowman's capsule. And then the middle one between them was the basement membrane. But how this ultrafiltration actually occurs? So this actually occurs because of differences in the water potential between the plasma in the glomerular capillaries here, and the filtrate which is there in the Bowman's capsule. So there is actually a difference in the water potential there. So usually what happens, water always flows from a region of higher water potential to a region of lower water potential. So what happens in this case is there are more solutes here in this case, because of proteins, they're red blood cells, so there are more solutes here. So actually the water potential is less in this case, but because of the capillaries, the pressure is more. So what happens is the pressure potential actually becomes greater. It outweighs the solute potential, and this causes the water to move out. So this is how more water can leak out of this glomerular, the plasma, right? And it can go into the lumen as a glomerular filtrate. So the rate at which the fluid filters from the blood in the glomerular capillaries into the Bowman's capsule is known as glomerular filtration rate. And in humans, this rate is about 125 centimeter cube per minute in both kidneys. So what makes the fluid filter through so quickly is actually the difference in the water potential between the plasma in the glomerular capillaries and the filtrate which is found in the Bowman's capsule. So inside the capillaries, in the glomerulus, the blood pressure is relatively high. This is because of the diameter of the efferent arteriole is wider, as compared to the efferent arteriole, so which is quite visible here as well. See, so look at the diameter of the afferent arteriole and look at how narrow the diameter of the efferent arteriole is. So this causes a buildup of pressure here in the glomerulus. So this tends to raise the water potential of the blood plasma above the water potential of the contents in the Bowman's capsule. So this happens because of this pressure difference here. However, when you come to think of it, the concentration of solutes in the blood plasma is more or higher as compared to the concentration of solutes here in the Bowman's capsule, right? This is because most of the contents of blood plasma actually here, they contain red blood cells, they contain proteins, so they're larger particles here. But if you compare that with the contents of the filtrate in the Bowman's capsule, they do not contain any red blood cells and protein particles. So this difference in the solid concentration in the blood capillaries as compared to the filtrate here in the Bowman's capsule, it actually makes the water potential here lower as compared to the water potential here in the filtrate. But the effect of the differences in the pressure actually outweighs the difference in solid concentration. So overall, the water potential of blood plasma in the glomerulus becomes very high as compared to the water potential here in the glomerular capsule here.
Glomerular filtration. Kidneys are bean-shaped organs which filters blood. It removes waste substances from the blood and regulates water and electrolyte concentrations within the body fluids. The end product of this function is urine, which is excreted outside of the body through urethra, containing wastes, excess water, and excess electrolytes. Urine formation involves glomerular filtration, tubular reabsorption, and tubular secretion. The nephron is kidney's functional unit. At the beginning of the nephron, the glomerulus is a network, tuft of capillaries that performs the first step of filtering the blood. The glomerulus is encased in thin, double-walled capsules called Bowman's capsule. The space inside the capsule and surrounding the glomerulus is called Bowman's space. As the blood travels through these capillaries of glomerulus, filtration causes a lot of plasma contents to spill out into Bowman's space through the glomerular filtration membrane. It consists of 3 layers of capillary valve: endothelium, basement membrane, and epithelial podocytes. This membrane allows some particles of blood to pass through, but not all. The fluid that is filtrate from the capillary blood into Bowman's space is called filtrate and forms the primary urine.